So you will receive different information of your interest, and you can select which one you want to go. It's all well organized sessions. Our goal is really just trying to enhance the educational and also information sharing within the healthcare society, especially related to the data mining, big data, or biostatistics community. So hopefully within this session you are enjoying and get some information or learning series from it. We are also actively seeking members or volunteers or new ideas if you want to share with us. And feel free to send email lower down here. And after the session, we always archive recording everything online in Dashu website. So you can easily find Dashu website from either Google it or find it later on in the slide deck. So next page, please. So this is Journal Club, as we mentioned. So we have a speaker, we welcome speakers and topics, and actually we usually have a monthly one, and usually the speakers, the one they presented from a very important high impact journal. Like last few months, we had one from Nature, and the coming month, March, we'll have different from different journals. But feel free if you want to volunteer your own work, and we can help you to have more visibility in the community of your work. This is definitely a great forum to present your work and share information with this community. We also welcome sponsors if you're interested to work together as collaborators in this journal club. Next page, please. So before I go to today's speaker, I just quickly publicize about March speaker. In the end of March, we'll also have a journal club. It's about cancer-related research therapy, about targeted cancer therapy, and how to predict clinical efficacy using general-scale signature of gene interaction. And Dr. Pen Zhang will present. He is working together with Shirley Liu's lab in Dana-Farber Cancer Institute and Harvard Public School. And the presentation they gave actually just recently published in 2018 Cell System. So it's definitely out of oven, very important research they have conducted. So after that being said, let's go to introduce our speaker today. I'm very honored to have Nancy Zhang, Professor Nancy Zhang, to join us today to present her research about statistical challenges and modeling in single-cell RNA sequencing. And Dr. Zhang actually is statistician over 13 years experience working in the field of genomics. She is currently associate professor in Department of Statistics, Wharton School University of Pennsylvania. And I'm very looking forward to Dr. Zhang's presentation today. So with that being said, I'll quickly turn it to Dr. Zhang, but just one thing to logistic to everybody. Please mute yourself. We'll have a Q&A session for about 10 to 15 minutes for you to ask questions or discussion. We'll record the whole session, so if you can mute yourself, will be very helpful. Thank you. So Dr. Zhang, please.
Okay, thanks, Sammy. Yeah, thank you for chairing this session, and I also want to thank Jing Lu, who helped make this a very smooth experience, and also the Dashu organizers for making this a wonderful platform for exchange. I'm very honored to be speaking today here. So I feel like I want to take this opportunity to tell you about single-cell RNA sequencing, which is a sort of a new technology that's been around for the last 5-6 years. And it's been offering a lot of new opportunities for both data scientists and biologists. So I'm going to take this opportunity to tell you about our efforts in making sense of some of this data and getting an initial understanding of this field. So I think some of you in the audience have worked in biology, so this might be a very basic knowledge, but just starting from the beginning. Okay, so biology is really about the study of cells, mostly, right? So cells are the basic operating units in biology, right? So, the basic paradigm is that each cell has a nucleus that contains its own DNA. and the DNA gets transcribed to RNA, which is viewed sort of as an intermediate product. And the RNA is then translated to proteins, and proteins are the machines that go and do their jobs. And so when we do RNA sequencing, we're sequencing this, this intermediate product, okay? And each cell really makes its own decision as to when to express certain genes and how much of that gene to express, okay? But before, I would say before 2010, most of the RNA sequencing that was done then was done on bulk cell populations. So what I mean by that is that all the cells are combined together, and you know, they're already together, but you know, we don't separate them, and we break open the cell wall, extract the RNA, which is a big pool mixed together from all the cells, and then we measure the average expression of every gene across all cells. Of course, that incurs lots of information, right? So what is it really the difference between bulk versus single cell RNA sequencing? When, you know, many years ago, when I first started learning biology, I remember that a common analogy that was given about cells is that the cells in our bodies are like building blocks, just like bricks are the building blocks of big buildings, right? So that was easy to interpret, but it's kind of unfortunate because what we know now is that our bodies, you know, our tissues are really, you know, not like a homogeneous brick building in the sense that the cells are very heterogeneous. So, for example, in this cartoon I'm showing here is of sort of a brain cell, brain tissue. And, you know, brain tissue doesn't just contain neurons, right? So it has neurons, it has microglia, which is a type of immune cell, it has astrocytes, and even within neurons, there are many different types of neurons, each specializing in its own task, right? So if we were to just measure the mean across this heterogeneous population, we'll be losing a lot of this fine detail. So I guess if we were to use a brick wall analogy, the brick wall would look something like this, right? Multimedia, right? And, you know, full of character. And that is what single cell sequencing is allowing us to see, right? So before going into the technical details about the single cell experiment and the data, I want to tease you a bit with opportunities that single cell RNA sequencing offers. Okay, so if you look at most single cell RNA sequencing papers that have been published, I think they are trying to address the question of getting a more detailed cell type classification of our tissues. So what that means is, you know, we know that, for example, the brain contains neurons, it has astrocytes, it has glial cells, but what other cell types are there in the brain, right? And among like neurons, what are the finer, can we get a finer classification of neurons? And so this is one of a very important question that that single cell RNA sequencing is obviously good for addressing. And oftentimes after we find the new cell types or the, you know, the better classification of cell types, we would want to find new marker genes for these cell types. So genes that are, say, expressed in these cell types but not in other cell types, which might be helpful for later on for us to do pull down and to isolate these cells and study them in more detail. And then also, here's another opportunity, which is we can use single cell RNA sequencing to understand how cells develop. As you know, every cell in our body came from one cell, the embryo, and there's a very complicated differentiation sort of timeline and trajectory in order for it to create all the tissues that we need. And how does this process happen? right? And what are the genes that are responsible at every checkpoint, right? So, so there's a lot of single cell studies trying to address this question. And it is a little different from the from the classification question that I listed as a first bullet point, because you can think, I mean, statistically, you can think of cell type classification as a discrete classification problem, trying to classify cells into discrete categories, whereas in cell differentiation and tissue development, we're looking more at a continuous differentiation timeline. So we're trying to find bifurcation points and things like that, which is, so fundamentally, it's a very different statistical problem as well. And then here's a different, the final bullet point I gave here is we can use single cell RNA sequencing to understand gene regulation. So, there's been quite a lot of studies looking at gene-gene correlation and trying to construct regulatory networks from bulk RNA sequencing, right? Looking at the mean expression across cells. But those have been, I don't think they've met with only very limited success. And I think the reason partly is because the decision of what genes to express is really done at a cell level. And when you smooth across cells, you lose a lot of signal. So if we have single cell, good high quality single cell data, it can allow us to see which two genes or which groups of genes coordinate at the cell level. And I think that is a very powerful method at trying to get at gene regulation. So there are many other opportunities. I'm just listing these few, I think, pretty representative, directions to go in single cell data. So I just want to give you one detailed motivating example that, sort of from a collaborator here at Penn. And I think this example is nice because we have a gold standard technology, which is RNA FISH. FISH stands for fluorescent in situ hybridization, which is another way of measuring RNA levels in single cells. It is much more laborious, but much more precise. So we're going to be coming back to this example a bit later to use the FISH data to validate some of our statistical estimates. So what this collaborator was trying to do was he was trying to figure out what leads to drug resistance in melanoma. Now, melanoma is a skin cancer, okay? So he started with melanoma cells in a dish. So this is a cell line, okay? And then he gave these cells a drug, vemurafenib, which is supposed to put these cells into arrest, so stop them from growing. Now, it was effective at first, so after he applied this drug, the cells stopped growing, but only for a while. So after 4 weeks, he noticed that these colonies start to form, and these are the drug-resistant colonies. And so when he, collect these cells, gather them together, and sequence the RNA from these colonies, he found that there's these resistance marker genes, which are listed here, that are highly expressed in these colonies. So he suspects that these genes are somehow responsible for the drug resistance of these colonies. Now, the interesting thing, this is a paper that was published in Science at the end of 2017. So the reason why this was really interesting was that this collaborator decided to go back to the time before drug was given and look at the original melanoma population and see what do the expression of these drug resistance markers look like before application of the drug. And what he found was that, so he looked at, I think, about 20-some genes, okay, which included on the right here, EGFR and NGFR, which are the drug-resistant genes. And he also looked at GAPDH, which is a well-known sort of housekeeping gene. And he found a fundamental difference in their cross-cell expression distributions. So GAPDH, as expected, has a sort of like a Poisson-like distribution, right? Sort of not very uniformly distributed, but quite evenly distributed across cells. Whereas these drug-resistant genes, EGFR and NGFR, have a very bursty distribution in a sense that there are many genes, many cells which are 0 or very lowly expressed for these genes, and there are a few cells that are jackpot, so they are highly expressed for these genes. So what they decided to do was to use a concept borrowed from economics, which is a Gini coefficient, to characterize each gene. So how much equality there is between cells. So for example, Gini coefficient of 0 are those genes that are like Norway, so they have perfect equality across cells. No gene is like that. And then there's Gini coefficient of 1, which is perfect inequality. So all the RNA copies are concentrated in one cell, and all the other cells are 0. So as you might expect, the genes sort of fall on a spectrum between 0 and 1. So what was interesting was that the housekeeping genes have sort of a low Gini coefficient because there's more equality, and the drug-resistant genes have a very high Gini coefficient. So it's very of high inequality. So this is one of the first studies which looked at sort of this burstiness of gene expression across cells as it would affect a phenotype, right? So phenotype being drug resistance. So this was interesting. So what he found later on, which I'm not going to go into in detail, is that they did a bunch of experiments to show that drug resistance in this line of melanoma is not related to anything like somatic mutations, which are known to be, clonal mutations, which are known to be factors in drug resistance in many different kinds of cancers. Okay, so it's not having anything to do with mutations in DNA. It is actually the stochasticity of RNA expression across cells that confer upon those lucky cells the genes and the materials that they need to sort of survive the drug, okay, and divide. So as you can see, this kind of analysis would not have been really possible if you were looking at mean expression across cells in bulk. So it highlights the fact that looking at the distribution of genes across cells, is burstiness, is dispersion, is Gini coefficient, is an important way of studying biology. So we'll come back to this in a bit. Okay, so I guess I was talking about the opportunities in single-cell RNA sequencing, but there are also, of course, a lot of challenges, and these challenges are really what give the computational scientists, I think, a lot of opportunity, a lot of things to do in this new field. So let's look at how the trend as to how the technology is developing. Okay, so a couple of years ago, when until 2016, we were still trying to keep track of all the papers all the key papers that were in the field, and obviously we stopped doing that. And now there's many papers almost every week. Okay, but if you look at this sort of small data set that we put together, right, of the papers that were published until 2017, I think. So here, this data point at 2009, this was the first single cell paper. It looked at 6 cells, okay, 6. And then, okay, the y-axis is in log scale. Okay, so you see that there is an exponential growth in terms of the degree of parallelization across cells, right? So the number of cells we are looking at per study is growing and growing and growing. And nowadays, I think it's not uncommon to have a study where there's millions of cells or at least tens of thousands. Okay, so if you look at, so that's good, right? We can look at more and more cells at the same time, but we want to keep the cost down, right? So the cost in these experiments is basically how many reads you sequence in total. And if you sequence more cells, that inevitably leads to a decrease in the coverage per cell if you have a fixed cost. So in the plot on the right, I'm showing the same papers, the same studies that are shown on the plot on the left, but here the y-axis is the coverage per cell, so the number of reads that are sequenced per cell. And you see that this is also decreasing, right, very rapidly. So the first study that sequenced 6 cells, each cell got over, I think, over 10,000 reads, okay? But nowadays, I think it is routine to do sequencing studies where every cell only gets a few thousand or maybe tens of thousands of reads, okay? 10, 20,000, that's quite common. And now it's also another trend is that more and more studies are starting to use UMIs. UMI stands for unique molecular identifiers. It is a barcoding strategy that reduces amplification noise. And I'll talk a bit more about UMIs later, but it's becoming more common because it makes the data more, I think, reduces batch effects and makes it more easy to interpret. So if the numbers, the blue numbers that I show on the plot on the right are the number of UMIs per cell. So you can think of that as basically how many molecules do we observe per cell. So you see that it's probably in the tens of thousands or thousands of molecules. But the question here we want to stop and ask ourselves is, how many reads is enough, right, to really profile a cell? How many UMIs, how many molecules are enough? Now the question is, how many in total, how many RNA molecules are in a cell, right? We don't really know the answer to that, right? That's part of the reason why we want to look at things at a single cell level, because we want to know what is sort of the RNA repertoire in each cell. But people have offered estimates, and we found this number, 200,000, from this review article published 5 years ago, and yeah, it differs depending on some people don't believe in this number, but that's sort of the order, 200,000. Okay, it depends also on the cell you're looking at, right? Nerve cells, for example, neurons are much larger, so they would contain more RNA molecules, and some other cells may be smaller, and you don't need as many reads. But you see that we are basically undersampling by quite a lot, right? So to profile 200,000 molecules, we're only sequencing about 17,000, right, or 20,000. So what that leads to is sort of a lot of missing data. Okay, so before going on and showing you the data, I want to also just briefly go through the experimental procedures that sort of generate the data. There's many different types of experimental procedures. There's at least 20 different protocols for single-cell RNA sequencing, but they sort of all follow these 4 basic steps. Okay, so the first step is that the cells need to be dissociated if they're in an intact tissue, or they need to be captured into their own individual compartments, right? So if you're using a microfluidic chip, then they are captured in their own little chambers on the chip. If you're using a sort of an oil droplet-based system, each cell goes into its own droplet. And then once it's captured in these little compartments, then the cells are broken open and the RNA is extracted. And then what needs to be done is that they need to be reverse transcribed into cDNA. The reason for doing that is that cDNA is much easier to work with. They can be amplified. Okay, so this reverse transcription step is critical. And then there's an amplification step where the cDNA must be amplified to an amount that allows it to be sequenced, because so that's a pre-amplification step. And finally, the sequencing step is kind of similar to the bulk RNA sequencing step. It's basically you take the amplified libraries and put it into Illumina or another sequencing machine. So there's a lot of details here that we're sweeping under the rug, but just looking through each step, what I want to highlight here is that there's loss at each step. Okay, for example, in the capture and lysis step, some RNA molecules are washed away and lost during cell lysis. And then Those that remain, some of them are not lucky enough to be converted to cDNA. And then if it were converting to cDNA, the amplification will reintroduce a lot of biases, okay? Fortunately, if you have this, you use this barcoding technique called UMIs, that really ameliorates this problem, okay? So what that does is it puts a barcode at the end of every cDNA that is unique to that molecule, so that in the end, if you get many copies of that molecule and you sequence them, you can just look at the barcode to know that they're from the same source, so that you, instead of counting the reads, you count the barcodes. And then finally, you know, after you have amplified everything, you put them all into a sequencer. And at this step, depending on how much money you have, you probably don't have the money to sequence everything. So sequence, you know, the sequencing step also is a sampling, sort of low coverage sampling. So a lot of the cDNA molecules are not sequenced. Okay, so what this leads to is that you have very sparse matrices, right? So in this matrix, every row is a gene and every column is a cell. Okay, so in these matrices, you would get a lot of zeros. Okay, so many of the data sets we look at have over 90% zeros. Okay, some of these are real zeros because we don't expect every gene to be expressed in every cell, right? So there are genes that are important for neuron function that you don't expect to see in skin cells. So things like that. But there are also a lot of zeros that are just due to technical noise. So maybe the gene was expressed, but not at a very high level, and it was dropped out during this procedure. Okay, so because of this, we have sort of formulated two challenges in single-cell RNA-seq data, two initial challenges, okay? The first challenge is that because we have very noisy data, lots of loss, we want to, is it possible to recover the expression distribution for a given gene across cells, okay? So we want to recover this distribution, okay? And the second challenge that we sort of put ahead of ourselves is to recover not just the distribution, but the actual true expression count for each cell. So for a given gene in a given cell, what is the actual count? You know, what you see is a noisy version of that count. So the expression distribution, let's go back to the original example that we were talking about for the melanoma drug resistance study, right? So remember this, everything in that paper, almost everything, was using FISH, fluorescent in situ hybridization, which is a better established technology, okay? So what it didn't say is that they actually also performed single-cell RNA sequencing data on the same population. Not the same cells, but they sampled from the same population and submitted those cells to RNA sequencing, single-cell RNA sequencing. And what they found was that they don't compare. So there's two technologies, FISH and DropSeq. And if you look at, and here in these plots, every little triangle is a gene, right? So for each gene, you can compute its Gini coefficient across cells in FISH, or its coefficient of variation or the non-zero fraction in FISH, and see whether the DropSeq version of that looks like the FISH version, because that's what you want, right? You want the same, because it's the same quantity, so you want to be able to reproduce that quantity using two technologies. And what they found was that the sequencing Gini coefficient grossly overestimates the FISH Gini coefficient. CV is the same thing, and the non-zero fraction, the sequencing, the single-cell sequencing, underestimates the non-zero fraction, which is what you expect, right? Because there's so much dropout, there's so much zeros created by technical noise that you just see much more zeros, and therefore the non-zero fraction is much lower. So, I'm just going to, I guess, spend 1 minute here to tell you about some work that I'm not going to give you details on. But I want to just say that we sort of formulated this. I'm going to spend most of my I guess, the rest of the half hour on the second problem of recovering the true expression counts for each cell. But for the expression distribution, I think it's also an interesting question, right? So here we just, we have to observe counts across cells. We want to sort of get the true expression level, the true distribution, underlying distribution. And with that, we can get these distribution measurements like Gini coefficient, CV, abundance of true zeros, and then we can use that in downstream analysis like cell type identification and transcriptional bursting and things like that. So that will allow us hopefully to get a better accuracy in these downstream analysis. So this is something that I think this is a problem that I think is important, and I guess that's what I'm going to say about that. I'm going to tell you about this second problem, which is recovering the cell-specific counts. Right? This is harder, right? Because if you have the cell-specific counts, then you automatically can get from it the expression distribution. But it requires more from the data in terms of quality. So what we mean by count recovery, okay? So let's say that there's this true matrix, which we don't observe, okay? Where, genes are the rows, cells are the columns. This is a true expression distribution, okay? And then what we do observe is sort of a lossy version of this matrix. So there's more zeros, okay? So here's a color bar. And what we want is to recover something that looks like the original matrix, okay? And we would like to, what we mean by good recovery, we can compute the correlation for each gene between the recovered estimates and the truth, and also for each cell. Now, of course, when you don't observe the truth, then you can't compute these things. But I'm going to show you a sort of a downsampling experiment that we do to try to get at these benchmarking measures. Okay, so I'll tell you a bit about our model now. It's a very simple model, I think. So our model is that there's a true expression which we don't know called lambda CG. C is for cell and G is for gene. And what we observe is YCG. And we assume, this is critical, we assume a noise model that you see on the left here, that YCG is Poisson with mean that is SC, a library size, times lambda CG. Okay, so what we are recovering is a relative expression, or you can think about it as a concentration. Okay, so this is a Poisson noise for UMI counts, and I'm going to show you in the next few slides why we think this error model works. And then, of course, we don't know this, so what we're going to do is we're going to use the informative genes, a set of informative genes, like genes that are in the same process, or, we're going to try to learn this set from the data to predict lambda CG. And the prediction we call mu, okay? So mu is our prediction for the true expression. But here is a caveat. There are genes which are very easy to predict. Maybe these genes are very predictable. Okay, they coordinate very tightly with other genes which have been sequenced to which have high expression. So their predictions are very good. But then there are other genes which are just more erratic. Okay, there are genes like that in our genome. They're much more bursty. They're erratic. So they're not predicted very well. They have high dispersion. So for every gene, we give it a parameter, a gene-specific parameter, phi g, that quantifies its predictability. Okay, so we assume that lambda CG is gamma distributed with mean mu and dispersion phi g. And we actually experimented with several different dispersion models. And I guess, I'm not going to go into details about that. But we sort of use a model selection criterion to find the best model for how to look at the dispersion. So, and then in terms of how you form this prediction, I think we did the very simple thing, which is we used GLMnet as an R package, and we just do a linear, L1 regularized linear log linear regression. okay, to form mu-CG. But we are working on right now using better methods to get at mu-CG, hopefully harnessing some of the machine learning tools that have been recently developed. So, but then I think the critical part is this Poisson noise model. Okay, so the question is, how do we check whether this noise model is true, is correct, right? So what we did a study using spiking genes. Okay, so I guess you could ask, how well do the spiking genes represent real genes? We sort of ignore that question for now because that's the best we have. So we're just going to use spiking genes to see whether technical noise follows this model. Okay, so the spiking genes are basically, you add these genes to each cell. These are fake genes. Okay, you add them to each cell at known concentrations, known and fixed concentrations, and then they go through the reverse transcription and amplifications and sequencing steps, just like the other parts of the transcriptome. Okay, so but the thing is, in each cell, when you spike in these molecules, these ERCC molecules, you can never be sure that you are spiking in exactly the number of molecules you want, right? Because this is first you dilute them, and then you take droplets of them and put them into each cell. So the number of molecules that end up in each cell is actually Poisson, with mean that that is the target quantity, but it is random, okay? So, and I think previous studies using ERCC spikings ignore this level of randomness. And then these genes, for these genes, you have the observed count, which is basically technical noise added to the original values. And what we do is we basically use our own tool, the deconvolution tool, to deconvolve using this error model, this Poisson error model, to get back at its original distribution, G hat. Okay, so and then we want to compare this G hat to what we think should be a Poisson distribution based on Brownian, dispersion, Brownian model of how molecules swim around in a dilution, right? So we compare it to what we think should be the ideal distribution, the Poisson distribution, and then we check to see whether it is Poisson. Whether it has the same CV as a Poisson, same gene as a Poisson. And then if this matches the Poisson distribution, then we say, okay, this error model must be reasonable, okay? Because if this error model doesn't capture all the technical noise in the experiment, then the distribution you get out of this will be more dispersed than a Poisson. And then so we looked at 9 publicly available ERCC spiking data sets. And these 9 data sets span a variety of single-cell technologies. Okay, Mars-seq, in-drop, drop. So if you do single-cell sequencing, this is a pretty representative set of the current technologies. Okay, and then we compared the estimated CV and Gini coefficients and non-zero fractions to what you would get if you had Poisson, and also to the negative binomial with a very small inflation factor, 0.015. And you see that for the second, the third, the fourth, fifth, sixth, and eighth, you get pretty good adherence to the Poisson distribution. The Markosko et al. Is the one that really stood out, and also I guess a little bit the Zeitman et al. Data. I think we have hypotheses for what's going on here. We think there's actually batch effects in their spikings. But anyways, what this is telling us is that at least for most of the data sets, the Poisson noise model is usable, I think. Okay, so the other reason why we want to use it is because it gives us analytical estimates, right, of our lambda CG. Okay, so based on this model, we can actually show that lambda hat CG, the maximum likelihood estimate, is a weighted sum between the data, between the data and the prediction. So you're sort of trusting the data and trusting the prediction, but not you're sort of adapting as to how much you want to take each of them, okay? So these weights depend on the cell-specific noise level and the gene-specific predictability. So for example, in this extreme case, where you have a very highly predictable gene, but the cells are sequenced at low efficiency, in that case, you would want to shrink very heavily towards the prediction because you don't trust the cells data. You want to trust the prediction more. And that's exactly what happens. So here you have the prediction on the horizontal axis and the actual lambda CG on the vertical axis. And you see that the black lines, the dots are the observed data, and you're shrinking towards the prediction, which is a red line. Okay, so your lambda hat CG is very close to the red line and very far from the observed data. And in the other scenario, the other extreme is where you have a gene that is where you don't trust the prediction, and but you have very high quality data. In this case, you just don't shrink very much. You pretty much just use the observed counts. Okay, and here I want to say that the 5G, the predictability for each gene is estimated adaptively from the data itself. So for each gene, we can figure out from, you can sort of fit a, by maximum likelihood, you can figure out what 5G should be. So of course, these are two extremes, and there are other scenarios where you have predictable gene and high efficiency, or unpredictable gene and low efficiency, and it's somewhere in between. Okay, so that's the model, and that's all the details I guess I have time for in terms of the technical details of fitting the model. I'll show you how it works on this original motivating example, this melanoma data set. Okay, so remember that in this data set, what they really cared about was the Gini coefficient, which quantifies how bursty the gene is across cells. And for this cell line, we have also, okay, in addition to the FISH data on 26 genes, we also have single cell RNA sequencing data. Okay, so between these two data sets, there are 16 genes overlap. What that means is there are 16 genes for which we have measurements in FISH as well as dropseq. Now remember, these measurements are not in the same cells, so we cannot compare cell level estimates, but we can compare each gene's expression distribution across cells and the gene-gene correlation. So any distribution-based measurements we can compare. Okay, so here is what I showed you earlier, which is the FISH Gini coefficient versus the raw dropseq Gini coefficient, and they don't really have, it's not very good. Here is what happens after you apply SABRE. So you see that you get things that sort of fall around the identity line, and so the Gini coefficients from SABRE look much more like those in FISH. And we also apply two other methods, MAGIC and SE-impute, which are also sort of there on bioRxiv. And you see that MAGIC really does not a very good job, and SE-impute is actually pretty good. Okay? And in addition to the Gini coefficients, you can also look at the actual distributions, right? The shape of the distributions. You see that, the gray lines are the FISH data, the black lines are the black solid lines are the dropseq. So you see that it's spiked around 0, shifted towards 0 more. And then after using SABRE, you get this dashed line, which looks very good. It looks like the FISH. And those are the two genes for which I think we really aced. But there are, there's a total of 17 genes that we did this on, and you see that for most genes, we get a substantial improvement. Okay? And how about gene-gene relationships? So pairwise relationships, right? So that is very important if we want to do things like construct gene-gene regulatory networks. And here are two genes that we know to be related, BAM1 and LMNA. And according to FISH, they are indeed highly correlated. That correlation is lost in dropseq, okay, because of the really high level of sparsity. And then if you apply SABRE, you recover the original expected correlation. Okay, and remember, SAVER is actually a Bayesian method, so it gives a posterior distribution for every gene, every cell. So the blue gradient is sort of like sample from that posterior distribution. The black is actually the posterior mean. You cannot just use a posterior mean because that will inflate the correlation. But we have worked out an analytical method to sort of scale it to get the unbiased estimates. And then here's just comparison. Here's MAGIC, and MAGIC really overestimates the correlation. So that is just one pairwise relationship. We have here all the pairwise relationships for all the genes in this data set. You see that when you look at the dropseq, it really dampens the correlation, right? The highly correlated genes in fish sort of don't have such high correlation in dropseq. We rescue those correlations. Okay, MAGIC inflates the correlations. SC-impute also doesn't seem to do well on this metric. Okay, so I guess I have 5 more minutes. I think what I'll do is I'll go quickly through this downsampling experiment. Okay, so there's something, the fish data is great because it's sort of a technology, using another technology to do validation. But it's not so great in the sense that you don't get measurements at the cell, they are basically of the same population, right? But not of the same cell. So you cannot see how well you're doing at the cell level, only at the distribution level. So what we did next was we did our own downsampling experiment. We took a high quality data set and we subsampled reads from that data set based on this Poisson error model. And then we sort of know this because we observe it, and then we generate this observed data. We recovered using a variety of algorithms, and then we computed the correlation to see how well we're doing. And then we did this for 4 starting data sets, okay? And then, so here is how well we're recovering the genes. Let me just show you all these methods, okay? So the black is for when you do nothing, okay? So you just look at the observed, and you see that the correlation with the reference is not great, okay? It's around less than 0.5 for most genes. When you apply SAVER, you sort of shift that up, okay? And here we are actually comparing to known imputation methods from bulk RNA-seq literature. So k-nearest neighbors, SED-based imputation, random forest, and they really don't do well for this problem. I think they're just not designed for the level of sparsity that we have. Okay, and I'll skip this because I don't think I have time. I just want to, okay, I think what I'll do is I'll skip all of this and I'll just show you one example of how this affects clustering, which is, I think, one of the most commonly done analyses on single-cell RNA-seq data. Okay, so so this is a data set that was published last year. Okay, they sequenced the mouse visual cortex, which contains a lot of different cell types. And what you're seeing here, these little clumps here, is it's called the t-SNE plot, distributed stochastic neighbor embedding. It's a type of dimension reduction, okay? So it takes a high dimensional single cell data and reduce it to a much lower dimension for you to visualize. And each dot on here is a cell, okay? So they form clusters, and these clusters correspond to cell types. Okay, so the cells within the cell type are clustered together, which is why biologists really love this type of plot. Okay, so t-SNE plot. And okay, so this is using just the raw observed data, and this is what you get using SAVER. Okay, so I guess to help you visualize it, let me just color them. Okay, so the color scheme here, red and blue and green, this is based on cell types that are identified by the original paper by very laborious manual exploration. So these basically experts looking at these cell types, zooming in and doing clustering and reclustering, and then they end up publishing these cell types. And you see that's why we like this data set, because there are these pretty trustworthy labels that we can use. So you see that these cell types in the original observed data are sort of mixed together, not as well separated as what you see here on the right after SAVOR. So SAVOR is able to sort of separate them and enhance the signal. Okay, and we are currently applying. We've applied this to quite a few data sets, and we are trying it on various problems, and hopefully, we can, it will have similar effects on other data sets. And then one common, just the last thing, one common thing that people worry about is, are we introducing spurious structure? Right, so we are sort of using all the genes to predict a single gene, and we're sort of averaging. Are we introducing structure that is not there? So we performed a permutation experiment where we sort of permuted the cells, so broke all the correlation between genes. And then after the permutation, we computed the pairwise correlation between genes to see whether there's any spurious correlation that is introduced. Now you see the observed correlations are around 0 as expected, because it's a null data set. We don't expect any correlation. And SAVOR actually shrunk them further towards 0, which is good. But other methods tend to, like MAGIC really introduces spurious correlation. So that's sort of a peace of mind. Okay, so finally, just to sum up, single-cell RNA-seq, we found a good noise model for this data. Our goal is to remove technical noise without introducing spurious structure. We have developed two methods, DESCEND for distribution recovery and SAVOR for recovery of single-cell expression concentrations. And we validated them using a variety of approaches. And hopefully this will help practitioners in their downstream analysis. And finally, I just want to thank Mo Wang, who worked on mostly, SAVOR was mostly Mo's work, and Jing Shu, who did the DESCEND software, and also the spike-in analysis that confirmed the noise model. And I want to thank our collaborators, especially Mingyao Li, who's a longtime collaborator on all of these projects, and also Arjun Raj, John Murray, Hannah, Eduardo, Roberto, who contributed the wet lab experiments. And if you're interested, here are the two papers on bioRxiv. Both are under review right now, and I'm actually under revision. So yeah, that's it. Thank you.
Thank you so much, Nancy. So Jing, maybe you can unmute the team, the whole attendance, for anyone want to ask questions. Can people hear me? I'm not sure whether he unmuted everyone yet. Okay. So thank you so much, Nancy. Anyone have a question to ask Nancy? We have a quick Q&A session. We have a few minutes. We can quickly discuss any burning questions to Nancy.
Big question here. Can you hear me?
Yeah. Yes.
Just quickly, the data, are you using the raw gene count, the UMI count, or do you do some normalization like FPKM, RPKM, or RSEM values?
We're just using the counts. You mean when we apply SAVOR or DESCEND, right? So we are just taking the counts and working off them. Yeah. But in some of our plots, like for example, when we are comparing when we are comparing, say, drop-seq genie versus fish genie and things like that, what we are comparing is the RPKM-derived genie versus fish.
So you use your algorithm to recover the true count and then convert to FPKM. Am I understand correctly? Our algorithm actually recovers something that is similar to RPKM, which is a sort of a relative expression that we're really recovering the relative expression level. So there's no conversion needed after we give our values. You can just use those values and treat them like RPKM values. Okay, thank you.
Yeah.
So here's another question.
Can you please get closer to the microphone? I think it's kind of hard to hear you.
Okay. So can you hear clear now?
Yeah.
It's better? Yeah, go ahead.
So my question is, you using a Poisson model to, for the Poisson model for the variation of the single cell RNA expression. But then you didn't tell us about what do you think about the differential expression. If we're using the model now, like IGR or BC, they're using MB model. So how does this
Okay.
together? Yeah.
Yeah, I think, okay, so there's this can be a pretty long discussion. So I guess we could correspond with email if I don't answer it very well here. So I think partly, okay, so Edge RNA-seq were designed for bulk RNA-seq, right? So I think they don't really take into consideration many of the technical aspects of single-cell RNA-seq. And in single-cell RNA-seq, there's I'm not saying that the data is Poisson. I'm saying that if you actually know the true expression of a gene in a cell, and also if you factor in the cell-to-cell differences in library size, in efficiency, and with those things factored in, they follow this Poisson model. Okay, so it's Poisson. The model is Poisson with
So this including both technical variations and biological variations?
No, the Poisson model is a model of only the technical variation.
Okay.
And it's only the technical variation once you've adjusted for the cell-specific efficiency. And this only works for UMI data.
Yes, okay, thank you, yeah. That's true.
Okay, maybe let's take one last question. If anyone have any question, I think it will be a good chance to have a interactive discussion here. Okay, if no one has question, I do actually have one quick last question. So Nancy, you talk about, very quickly you touched base on the traditional missing data methods comparison with yours about KN SVD based imputation.
Yeah.
And you mentioned it doesn't work well at all from your graphic display.
Right.
And you kind of mentioned about the structure of it. But can you touch base more on what's your understanding why those does not work well for single cell RNA data?
Yeah, so here is the comparison, right? And see these cluster of green and brown curves are on the left here. They don't work very well. I think there's several reasons. One is this data that we have is not just, these methods work well when there are some zeros in your data, but it's not too sparse, and the data is really very, very sparse. And also, these methods don't assume that there's real zeros in your data, right? It assumes basically if you don't see a data, if there's a zero, it's missing, and it's not. In our data, there's both missing due to randomness, missing due to the noise, and also just biological structure zeros. And also, finally, these methods, I think, just over-smooth. I think that's the reason why they don't do well, because they over-smooth. They introduce spurious structure. And a single-cell data is not really, the cells are erratic. Okay, cells don't really lie on a low-dimensional subspace. So the SVD assumptions are incorrect. So
Okay.
I think at least those are the things.
Yeah, Thank you so much. I think we are about the time. I want to really thank our audiences and also thank our speaker, Nancy. Just 2 points here is a really interactive and a very informative session. We received comments from the audience from Freeman. And also there's one audience is asking if possible to have a copy of the slides. So as I mentioned in the opening of the session, we will archive the recorded session on DASHU website. And I think if you receive this information or session from the DASHU email list, as you are one of the members, we'll email the link and everything after the session archiving is finished on the DASHU website. Should shortly receiving that email notification. But if you're not a membership, you want to still receive the slides or archive recorded session, you can shoot an email to DASHU's email at the beginning of the slide. So maybe Nancy, can you go back to the first page so people can quickly see the contact information? If anyone interested to go to additional session in the future or want to be part of the email list to understand our sessions, you can contact DASHU on the bottom line here, dashuinfo@gmail.com, and you will become a free membership. And also we want to promote, feel free to reach out to us if you want to have a presentation here and you have a good journal published research. We're happy to provide our platform for you to communicate to the community and share your research and work. Okay. As that being said, thank you guys so much. Thank you, Nancy, for joining us, and I really appreciate your presentation and contribution here. Thank you, guys. Have a good rest of your day.
Thank you.
Okay. Thank you. Bye.
Bye.